Actually, fair warning, next one will be a move. Now, just so you're put up your hand if I all of a sudden switch and you didn't have time to write everything down, because I am not going to write everything down. I am going to use the writing that's already on the board because I am feeling ill. I'm pretty sure I have a fever. The last analogy I used involved Texans, people in Brentwood dress, and a military squad all killing each other. And so my thinking, probably not the best. I'll try to come up with a better analogy for this time. But we're going to start our biology unit today. I'm going to give you some time at the end of class to grab your lab, go through the questions, ask me questions. I have a correction to do and a section on the data booklet to show you. And then you're going to keep your lab book and you're going to bring it to next day, in which you're going to have a quiz, and it's open lab book. In the lab book, you should have the marking sheet. You should have a copy of the lab sheet and the procedure, and I have new copies if you lost that, and all your answers to the questions and your observations, and the questions that you get on the quiz will be based on that. It's still, you can't talk to anybody else, you can't use the internet, but you can use your lab book. We are going to start today on DNA. Your ATCGs is a reference to the code of DNA. ATCG are the nitrogenous bases of DNA. That goes through how are we going to get all of that code and all that instruction that makes us us into your DNA to grow a human from scratch. CRISPR, I've been saying for years, is coming, and it's going to be a very interesting world once it does. We're in the spot when like the first supercomputers were, and people were looking ahead and trying to predict what the world would be like, and now we all have a laptop and there's personal computers and the entire internet. CRISPR's similar in the biological field. We are going to be able to do some amazing things, and I now can officially announce it's not coming, it is physically here. We have done our first human trials of curing sickle cell anemia with CRISPR. If you don't know what CRISPR is, yes?
I was going to ask what CRISPR
is a mechanism that we have borrowed from viruses in order to cure genetic diseases. So for example, we've had genetic therapy for a long time. How many of you have heard of boy in the bubble disease? And I only phrase it that way, one person. What is boy in the bubble disease? Allergic, yeah, to the environment. You have essentially no immune system. You get a cold and it is life-threatening. You can't fight off bacteria, you can't fight off viruses, your immune system is so suppressed. I say boy in the bubble because there was a comedy, quote unquote, that was made and it was called Boy in the Bubble. But it's obviously attacks people of both genders. Essentially, you live in a special room, all your air is filtered, so there's no bacteria and viruses. All the food you were fed is sterilized, so that you are not exposed to any viruses or bacteria. Your parents pick you up and hug you through plastic and gloves or wash their hands very, very well. Because if you get any infection at all, it is life-threatening. You do not fight it off and your immune system is so damaged. It is a one point mutation that causes that, and it causes that cascading failure in the immune system. Your whole immune system doesn't work because of one mutation. We've had genetic therapy for a long time, but it hasn't worked very well. Back in the 90s, we actually tried, I say we, humanity, and it was actually French researchers were trying a new form of genetic therapy to cure this immune disease. And there was also a rather famous personality who was a teenager and who said, when I turn 18, this made the papers in France, they were a person that was suffering from this immune disease, and they said, I'm coming out of my room, I'm removing the bubble, I'm going to live my life like everybody else because I am tired of living this way. And I don't care. If I die in 2 months from disease, at least I was living for those 2 months. And the scientist said, well, since you're going to do it anyway, would you like to volunteer for our experiment? So he, along with 7 others, volunteered to be cured in this way. And he lived for 2 years and then died of leukemia. But during those 2 years, he was disease-free, or at least like everybody else. He got a cold, he fought it off, his immune system worked fine. Leukemia in humans is not a transmissible disease, so he did not die from catching something. But he did die from the cure. Because when you introduce mutations into someone, which is what a genetic cure is, you're putting DNA into your DNA. So you have to put it somewhere. And wherever you put it might be interrupting code that's already there. If you're interrupting code that's already there, well then you could have another side effect, and his side effect was leukemia. Of the 8 people they tried to cure, 2 are still alive today. Now they're alive today and completely cured. It just happened to get into just the right spots. They never got leukemia, they never got any other form of cancer. They were very lucky. But 2 out of 8 is not a great result in terms of medical therapy. And so that is obviously, it would never went further for human testing. But it did show it could be done. CRISPR allows you to be very precise where you put the genes. So instead of putting it into the genome randomly and maybe creating a mutation, you can say, we're going to put it exactly here where it won't bother anything, it won't cause a mutation that gives you cancer, and now you have the genes. Sickle cell, does anyone know what sickle cell anemia? Particular mutation in the hemoglobin of your blood. The hemoglobin of your red blood cells are filled with sacks of hemoglobin, and they should look like this. It's not a donut, it's not a hole, but it does have a dimple. And obviously it's red and it's full of hemoglobin. In people with sickle cell anemia, it collapses and it looks like this. And so that's the red blood cell, and it collapses and it literally looks like a sickle. And then these edges catch on the side of your vascular tissue, your arteries and veins, and they can cause clots, and it is life-threatening. It requires a fair amount of therapy. It is stuck around, probably because it provides some protection against malaria, and there's variations of this disease all over the world that have persisted in areas where malaria is a going concern. Because if you have that, you won't die of malaria when you're very young, but you will have troubles once you hit your 30s, so it's not great for longevity. But it is due to 1-point mutation. CRISPR, you can get in there, fix the mutation, and now it's producing regular hemoglobin. If you have a point mutation for diabetes, theoretically that means that type of diabetes is curable. If you have phenylketonuria, where you can't digest a certain amino acid, and you must avoid all plants and animals protein because it has that amino acid in it, and you have to eat a special mix that is essentially made in the lab. 1-point mutation, curable. It is going to be a very big deal because we can now cure the things, but now we can change anything. Drops in your eyes and change your eye color. Yeah.
Like, so you could could you adjust physical attributes too, into the future as well?
You get a genetic readout if you're developing embryo, and you go, the genes for tallness are where I'd like them. Let's make them over 6 foot. Let's give them this eye color. They've got a this % chance of red hair, black hair. Well, let's go with this and this. And take out these diseases, but add in these pieces for long legs because I'm a rower and I want this particular ratio because I want my children to be rowers. Now, if you've ever not wanted to do what it is your parents had planned for you, imagine they had the ability to make your physical structure exactly what they planned for you. So there are all kinds of ethical problems with this too. And there is no perfect human with just the right genes. A lot of genes are successful in certain environments and unsuccessful in other environments. Sickle cell anemia, if there's an area with high malaria and you don't have access to medication, that is actually quite a good gene to have. And there's a ton of genes that are good in all sorts of ways. Like, yeah, this might increase your risk of cancer slightly, but it lowers your risk of diabetes in this area. And what kind of food you eat over your lifetime may actually make a difference as to which one's good or bad. And it's not as easy as cheetahs are fast, so let's take the genes out of a cheetah and put it in a human. And then you have a human with little tiny spindly cheetah legs that just flops around the floor and can't run fast because genes are meant to go with the whole organism. It's not just like fast from cheetah, it's not like superhero comics where you absorb their essence. So it's complex, but they'll start with the low-hanging fruit. 1-point mutation differences. Small mutations that make a big difference are probably curable in the next little while. Yeah.
How do they like implant these like mutations? Do they use like stick something into your body?
Yeah. So viruses, really good question. So we haven't gone the details of DNA, but you have a code. And the code is on 2 sorts. Little preview. Does anybody notice a pattern? Yeah. So if it's an A, it's a T. If it's a T, it's an A. If it's a C, it's a G. If it's G, it's a C, and vice versa all the way through. And so a virus invented a mechanism where it goes in and it breaks apart the code. Literally goes. And it puts in its own bit of code. And then forever after, that code is in there. So it literally inserts a gene. You could do it other ways. Like if you're like a backyard biochemist and you're doing your own genetic engineering, they've done it with plants with a shotgun. So you take your plant samples, you take a shotgun, and you load it with the DNA you want to inject in it, and you shoot the plant. And then you just grow bits of the plant to see if the gene actually went in. And sometimes, by purest chance, it does. It kind of only works with plants. If you shoot animals with a shotgun, it doesn't really work in the same way. Yeah, it's not like you go around shooting genes in people. Although with CRISPR, maybe, because now we think about, we're now using a mechanism to insert genes into people. Imagine like you could soak the virus into your skin and give yourself luciferase gene, which would give you a tattoo that glows in the dark. No ink, just changes the production of your cells. Or you could develop a virus that spread with a cold that gave that to everybody. There's all sorts of problems and benefits that could or might happen that we are a little bit worried about and also really excited about. There are so many diseases that we know where the problem is. We just don't have a small enough wrench to go in and fix it. CRISPR gives us a small enough wrench. We can now go in and start splicing things. The fact there's our first human trial now, we are on our way. It's really exciting, a little worrying, but also really exciting. And a little worrying. So, that's what we're doing in terms of biology. Pull up next page of your notes. We are going to do some notes and give you a little bit of background. Questions?
Can you make cough syrup?
Can you make cough syrup?
Yeah, like can you? Like do you know how to make cough syrup?
What? Yeah, well, why wouldn't I just go to the store and buy cough syrup? And if I wanted to make meth, I'd just go buy the cough syrup.
Well, like, let's go. Like, do you know?
Our local drugstore had to hide the cough syrup every regatta. Well, because you're not allowed to sell it in the States because people take it and use it to make meth. So as soon as people saw that our version of that particular type of cough syrup were on the shelves, somebody in the groups from the States bought it so they could take it back across the border and sell it to people who wanted to turn it to meth, presumably, was the guess. So they had to pull it off the shelves while the Americans were here.
What if they just sort of
But cough syrup is like one chemical tweak away from meth, which is why meth heads can make meth. It's not a hard synthesis. But I'm not going to spend the class talking about how to make meth.
One more question.
Is it DNA related?
It is.
Okay.
It has
I'm
to do
trusting
with
you
what
now.
we're just talking.
Pardon?
It has to do with that. I have a question. Can like, as an average student go buy said cough syrup to use it for the genetic stuff?
I can't think of a situation in which you needed to, and of course the average student wouldn't, because the average student is a good person who wouldn't make meth to contribute to the downfall of our society. So, we will continue on from here. I'm stop talking about cough syrup. Anders,
Can
Focus.
you tell us how to make meth?
nope.
Wait.
If you want to know how to make any of those, there's literally YouTube videos on it. And one, two.
Alright, how can we make it?
Remember, the reason you don't make it is the same reason you don't put poison in somebody's food. All of you have access to Thrifties, and all of you have access to all sorts of chemicals and could do a whole bunch of horrible things, and you don't, because you decided today to be good people. Congratulations, give yourself a pat on the back.
What's something called phenol?
And I'm certainly not going to do or say anything to encourage you to be bad people. Okay. Yes?
What's that like next to studying, like after studying?
Oh, sure. Yeah, I'm going to do that right now. Now, number one, this is not a history type course where I want you to memorize the dates, but I do want you to know the concepts and what happened. And the dates are there specifically to give you a sense of time so that you know roughly when. So it's not like it was yesterday or last week or 100 years ago. Could make a fairly big difference. 1869, a chemist, John Friedrich Meischer, was studying pus in medical bandages. So, people return from the war, their wounds are oozing, they have medical bandages full of pus, and Dr. Meischer goes, that is a useful area for research. And he discovers DNA in the pus. We don't know at this point, and we, I mean the scientific community and humanity, we don't know what DNA is used for. We don't know what it is. We know its rough structure. We know it's a deoxyribonucleic acid. So we know it contains like a ribose, a base, a sugar.
Is there going to be an S in there?
Wait,
An S in where?
Deoxy.
Nope, deoxyribose. The S is at the end, just before the E.
wait.
Oh, maybe it's in German.
That's
Does it say?
is that nuclear or
Nucleic. Deoxyribose nucleic acid. Oh, look. The computer got it first try. Yeah. Yes.
Is for the, under the deoxy
Ribose.
in the brackets, is that an underline and arrow or?
That's an underline and arrow to point out that the ribose, the os in this case, whenever you see a biochemistry word and it ends in os, it means sugar. So what is glucose? Is
sugar?
sugar. The sugar on your table is sucrose. Sugar high in molasses, maltose. Sugar in fruit, fructose. You've heard of high fructose corn syrup. That's just sugar from fruit. The reason they don't like high fructose corn syrup is it turns out lots of sugar is bad for you. And the type of sugar that a lot of people eat in the studies that they're looking at have really high fructose. If you switched to sugar that was low fructose and you kept eating the same amount of sugar, would it be better? Well, maybe a little,
What's
according to
like
some studies. But really, you don't need to have a lot of sugar.
What's
Just
like the least unhealthy sugar?
one that's in a fruit.
So sugar.
So eat something, like most things, if you take the pure form and eat only it, your body wasn't designed for that. Your body was designed to eat things together. So it's expecting fiber, it's expecting other proteins with it. You eat a piece of fruit, it has all of there, the absorption goes differently. If you eat a tablespoon of sugar, then you have none of that and it goes straight to your bloodstream and spikes your insulin. It's not nearly as good. But we evolved the ability to detect sugar because that meant high energy and lots of vitamins. We can't taste vitamins. We taste the sugar to get the vitamins. And then cleverly we went, well, we like the sugar, so we'll take the sugar out of the vitamins and invent chocolate bars and gummy bears. And now we get the sugar without the vitamins. So that's why it's meant. If you eat it in its original food, and that doesn't mean uncooked or anything, that just means eat fruit if you like sugar. Casual? What?
Oh,
Caused. I will repeat all the words on there. Now, notice 1869, 1990s. This is about 100 years later. We know this. We know the rough structure. It's deoxyribonucleic acid. So we know what makes it up, but we don't know what it does. And I'm going to go through this study in a little bit, or a version of this study, a little bit in detail. It showed that DNA caused bacteria to change their behavior. And the one that we're going to look at specifically, bacteria would kill and infect a mouse, or they would infect a mouse without killing it. And these experiments showed that the DNA was responsible for that, no other type of molecule. This allowed viruses to infect cells, and this showed there's something special going on with DNA that wasn't going on with other things. Up until now, they've been guessing that protein, with its varied forms, is responsible for all the information, but this was suggesting DNA. 1953, Watson and Crick discovered the structure of DNA. And I put it in air quotes because Rosalind Franklin should definitely be on that list. It was her work and her discovery that really made the breakthrough. And Watson and Crick stole her pictures and didn't credit her for the pictures, nor credit her in her paper. And in terms of scientific standards, they absolutely should have. The only reason she wasn't added in retrospect is that she had died, and they don't award Nobel Prizes to dead people. So you can't go back in time and award a Nobel Prize to Einstein. All Nobel Prizes are only given to living people. And they won't even do it to correct a mistake, if a mistake was made. To explain what happened, there's another picture underneath this one. Yep. Oh, if you need to go, you can go. This is on the right, Watson and Crick with their original model of DNA. I would like to get a model of DNA. There's a 7-foot tall one made out of molecules like we have here when I make little molecules. You may have seen in the background of Big Bang Theory, if you ever watched that TV show. It's about $3,000 to buy, about $1,000 shipping. So, you know, if your parents ever want to donate some money, specifically say they can only spend it on Mr. Neufeld science class. And then I'll get that model. Pardon?
How much is it?
It's like $4,000 with shipping. It's tall. However, I will try to convince Ms. Allen a different way, in which I will say, look at our beautiful new space. The beautiful science labs. You know what would make this go really well? A sculpture of DNA. It just screams science. So, this next question requires you to put up your hand. How many of you think that your education would be improved with a 7-foot tall DNA model? It's required.
You should.
Excellent. I'll tell Ms. Allen that everybody agreed.
You should get a Kenyan yoga.
And Pablo didn't, so I'm going to make his
No, I
mark go
didn't.
down.
That's-
Mark go
that's
down.
wrong. That's recorded, right?
Pardon?
That was recorded, right?
Yes. Awkward. Yes. So, Watson and Crick.
Okay.
Watson and Crick also didn't start off the discovery. Linus Pauling was a chemist who was starting on the structure of DNA. He had actually made these models of nucleotides, and he was trying to fit them together to try to figure out the structure. And he eventually got so frustrated he gave up. And then Watson and Crick would like, hey, we're working on that. Can we borrow your models? Linus Pauling, being a scientist, and you share things, so he took it off and gave it to them, and he shared them. So that he had already made some progress. Linus Pauling is the only Nobel Prize winner who has won 2 Nobel Prizes unshared, one for chemistry and one for peace. He did a lot of the work in electrochemistry on electronegativity in chemistry. And he did a lot of work in negotiations and talking to international communities, particularly for peace. So there's his award there. He also went alternative health crazy at the end of his life, and he's responsible for the whole, you know how when you have a cold you should eat vitamin C?
Yeah.
Yeah, that's all from him. That's not really true. Every study has shown that if you eat lots of vitamin C, you don't necessarily decrease your cold at all. But he was a big proponent of massive doses of vitamin C curing everything, from cancer to everything else. They did find out that it can have some bad side effects, because if you crack your teeth on that many hard, chewable vitamin C's, you can actually literally crack your teeth. And it turns out if you take massive doses of antioxidants, they get so good at pulling off electrons from things, they'll pull it off of your DNA and give you mutations in cancer. So it turns out super high mega doses can actually cause cancer. Certainly, there's no benefit unless you're vitamin C deficient. You don't need to eat pills. Eating oranges, however, are healthy. Yes.
I was gonna say, if you're sick and you eat like lots of oranges and drink orange juice, does it help?
No. You're sick. It won't make a difference.
Just
If you are vitamin C deficient, like severely deficient, certainly you might get sick a little bit more often. If you're getting things like vitamin C deficiency, scurvy, or lack of vitamin D, rickets, like all these things, yeah, you need some pills and you need to do. But if you don't have any of those and you're getting then eating a pill, almost universally, every study has shown pills do very little.
Wait,
You
so-
need to get your vitamins in food.
So if
Pills do one thing really well though. They make very expensive pee. Yeah, go.
So if you're sick, like you can't eat anything to help. You just
No.
have to wait.
Eat well and yeah, get some sleep. Sleep is the number one thing. There and then there.
Did the vitamin C guy, did he die from getting mutation?
No, no.
Or
And
just-
it's a rare thing. It's like you're like, oh my god, I've been taking vitamin C pills all my life. I'm gonna get cancer. No, we're talking mega doses. Because after him, some people started injecting like large amounts of vitamin C to the point like you couldn't even eat that much. We found it out in them, and all we found out is it did induce mutations, which could theoretically cause cancer. I don't think we even found a cancer that actually was caused by it. It just caused some mutations. Was there another? Yeah.
Yeah, I was curious, like, what's like the length that we can go with mutations? Can we like get like superpowers?
I mean, technically, if you live in an area with malaria, sickle cell anemia is a superpower. You probably have 2 to 3 mutations that are different from your parents. Everybody does. Most of them are silent and they don't do anything. You could have a mutation that makes you slightly better at something
Could you inject
Um,
like everything into you?
and die.
No, like everything that's good, like
If you like, what do you mean by inject? Inject
or,
what?
All the yummy genes.
the good genes. Could you
Oh,
inject the good DNA?
well, again, what's a good gene? If you want fast speed and you decide, oh, cheetah genes. Okay, we can't do that because then we'll have cheetah legs and we'll flop around. But like, put it another way, you have fast twitch and slow twitch muscles, and the ratio of those have really shown that make a difference in which sport you're playing. Fast twitch muscles, soccer. You want fast energy, moving around, dodging quickly. Slow twitch muscles, weightlifting. You can will lift far more heavier loads than if you have slow twitch versus fast twitch muscles. And what ratio you have is controlled by both genetics and your training. And people who are born with a high level of slow twitch muscles, there's certain actual families that have a particular genetic disorder that just gives them an abnormally high amount of slow twitch muscles, and they have the most number of Olympic medalists for weightlifting as well.
Just that
Because
one family.
yeah, that one family. They also have a lot of members of their family that are in wheelchairs because their slow twitch muscles don't allow them to walk properly. So is it a good gene? In some people it worked, but in some people it didn't. It's actually considered a metabolic abnormality. You could live a perfectly normal life and just have big muscles, or you could actually not be able to walk properly. So it's a lot more complicated than just the good genes. Yeah, you want to finish off the thoughts? And then
So would it be better to have fast twitch, more fast twitch, or more?
it turns out if you're just a regular person, it's good to have a good mix of both. And you can do both.
Okay.
If you're aiming to be the world's best soccer player, having a higher ratio of fast twitch would be good. But that involves a whole host of other things. And do you really want to have that extreme level if it ends up you're not the world famous soccer player?
Like, could you like delete like the code that like limits your like muscle growth? So could you like in theory be like super jacked?
Yes, but again, it's a genetic disease. There's a there's a strain of cows that have a particular mutation that makes them look super muscular all the time. I'll see if I can get some pictures. They're on a particular farm, they have this mutation. There's a family of humans that have that particular mutation. They also have a really high number of weightlifting medals in the Olympics. And a lot of them have other problems with their heart doesn't grow properly and they die young. And so genes have consequences beyond just one area. So not always the best. Anyway, go back to the story of DNA. They're trying to make the models from Linus Pauling. They can't do it. They don't know what the structure looks like and they can't quite figure out how it's put together. What they're missing is they're looking for a single molecule, and DNA we know at this time is double-stranded, and they haven't thought of the double strand. That was a key piece of information that Rosalind Franklin figured out. She was also working on the structure of DNA, and she was taking pictures of it with X-ray chromatography. This is an X-ray picture. Now, without going into details and math, all I can say is that picture tells you it's a double helix. This is like giving you the answers on the test. This tells you what the structure must be. With that knowledge, you now can go back and do it. They took her pictures without her permission. She didn't even know they were going to use it. The first time she saw it was when they published their paper. I have an image of my head because when you're affixing this to the X-ray machine, DNA is very elastic in its pure form and kind of stretchy, and so she'd have to stretch it on two very thin needles and then do the X-ray chromatography, and often boing, pop off like an elastic band, and then she'd be crawling around on her hands and knees looking for it. And so I have this image of like Watson and Crick. She's crawling around looking for her DNA, and they're like sneaking into her lab to steal her pictures. Because they literally did, but in retrospect, they probably just, they worked in the same place, grabbed their staff keys and walked in and took it. So her name belongs in there with theirs. It was her picture that told them it was double helix. They went back to their models, built it, published the paper, and now we know the overall structure. Question? You had a hand? I am going to go through the experiment now, and the experiment needs some explaining. Okay, turn the page.
Are we gonna like kill a rat?
We are not going to kill any rats. No. And I'm also this time not going to mention any tests.
Anyway.
Never mind, I just mentioned it. Okay. We have a rough strain of bacteria. We take that bacteria, we inject it into the mouse, give the mouse an infection. The mouse doesn't die. That's what the word non-virulent means. Doesn't mean it doesn't infect, it means it's not lethal. So the word under there is not lethal. Any questions? Write those in. Then, and strain refers to bacteria. We have another strain of bacteria. It's called a smooth strain. It is virulent, and it is lethal. So we take the smooth strain, we inject it into mice, and it kills the mice. These are normal outcomes that we would get with the rough and smooth strain. Rough and smooth literally refers to their appearance and the types of colonies they make. When they make colonies, smooth strains literally look smooth and rough strains look rough. There's nothing beyond it there. Okay? So we have a strain of bacteria that kill the mice. Sorry. And a strain of bacteria that don't kill the mice. Good. These aren't really experiments. These are just set you up. Any questions? Excellent. Now, we take our red bacteria and we heat them up and we kill every one of them. So we essentially put them in an autoclave or a stovetop or whatever it is, microwave, and we kill them. And if you take the dead, smooth strain, virulent bacteria and we inject it into mice, the mice live.
Wait, so I want to say that
Virulent.
Does that mean just it is
Lethal. Yeah. Virulent means lethal.
What does it say above that? Like controls?
Pardon?
Smooth is wait.
Smooth? Oh, both of these are what we would call controls. We were showing what happens under normal conditions. We haven't changed anything. So this is a control in terms of the experiment. This is a control. And then here's our first experiment. is the thing that kills the mice something that's killed by heat? And it is. Whatever it is that kills them is part of the living organism, because dead ones don't infect and are not virulent. This is not true of everything. One of the reasons you avoid red tide is because the toxins produced by the things in the water, even if you heat everything up to a certain temperature, those toxins are still there and they'll still kill you. You're not being killed by the living bacteria, you're being killed by their very dead toxins.
Question, is that a fire alarm?
No, no, that's just a thing moving around.
Totally happy with it.
Second question, do you have fun making these?
No, I'm literally summarizing an experiment. I got this from an explanation of an experiment. If you think, having seen my drawings, that I drew this, I am very complimented. No, someone else made this. So, took this, heated up in a microwave, don't die. I'm going to come up with my analogy now, so let's not use the analogy I used last times. This is our Science 10 class. And the lethal strain, let's see, who could take you out, is let's go with a group of what is, what's that group called? It's a Canadian group. No, CSIS is the organization.
Yeah, I was just thinking of like how many groups that was just came to mind.
It's the equivalent of the Canadian Special Forces.
Oh.
I'm trying to remember what they're called because right. The Lethal Joint Task Force 2. These are heavily armed Joint Task Force 2. These are the Canadian Special Forces. So they burst into here and we all die. That would be expected.
I don't think I would.
Except for Julia.
Okay.
These here, these are the grade 8s. They burst in and they die. Okay? Now we take Joint Task Force 2. Pablo, you keep turning around. And we heat kill them. So they're just lying there with all their uniforms and guns and everything. And they all get wheeled into the classroom, and we look at them, and they do nothing, and we go, yeah, it's not going to be a problem. Then nothing happens. Okay, clear? Okay. Not the greatest analogy, but a little better than my last one. Now, yeah?
What's the third one?
Heat killed smooth strain.
What's the thing under it?
Oh, that's to show the body of Joint Task Force 2. They've been heat killed, so they're dead. Mm. Again, ill, possible fever. My analogies may not be the best, so I am asking you to put up with it. Okay, are we good though?
Yeah.
Okay, now we have the live grade 8s. That's not a good pen color for this. What would be good here? So there's the grade 8s. We have the heat killed Joint Task Force 2. So both of them together, the live grade 8s are with the bodies of Joint Task Force 2, and they burst in here and they discover that they killed the Science 10 class. What happened? They took their weapons. See, this is why I explained this without that analogy, and nobody got it. As soon as I put the weapons in there, even though it's not a good analogy, it worked. The grade 8s burst in with like weapons that are a little too heavy, but still weapons. So what did the rough strain bacteria get from the dead smooth strain?
They got
Yes?
yeah.
Yeah, deadly stuff. They got their weapons, whatever they are. At this point of the experiment, we don't know what it is, but we do know that it's something they got from them, that you need someone alive to use, because the dead ones don't do anything, but they must have gotten something from their bodies. And we don't know what they got from the bacteria bodies, but it must be something. Is that good? Excellent. Because then the last one is what tells us what it was. And I'm only presenting the last one because the last one is the one that was successful. There's actually a bunch of other ones. Because what they would do is they would, okay, let's give them the heat-killed versions, but like, I'll put it in the same analogy, we cut up all their underwear. They're like, oh, the grade 8 still killed them. I guess it wasn't the underwear. And we took them and we cut up all their vests. It wasn't the vests. And then we took them and we cut up all their guns into little tiny pieces. And now, suddenly, it doesn't work. Must have been the guns. We took the heat-killed smooth train and we chopped up the DNA. And then the mouse survived. So we knew that the weapon, whatever it was, the information, the instructions, whatever it was the DNA. And this told us that DNA was really important for instructions.
What does it say under chopping? Is it enzyme?
Enzyme, yeah. DNA chopping enzyme. Woo-hoo! It recognized it. Sorry, got excited. Pardon?
I'm to be trying to text right now.
You're trying to?
You accidentally selected the whole thing, I think. So what would happen if you did now?
It selected the whole thing because the whole thing was grouped. I'd have to go back in, ungroup it, and take away too long. So, this experiment told us that DNA was important. For a time, they thought DNA was just a catalyst or something. So DNA is important. It passes information. It can change the behavior of things. And that let us know that things were hugely important with DNA, and then led us to the first structure. So, starting on structure. It is extremely large. If you took the DNA out of one of your cells and unwound it and laid it out as a single double-stranded, it would be 2 meters. I happen to know that my arm's length is about 2 meters, 1.8, 1.9, if I bend my fingers. So, one cell. If you took the DNA out of all of your cells and laid it end to end, you would die. Because you wouldn't have any DNA in your cells.
Well, so
Like, that'd be bad.
all your cells be destroyed.
Yeah.
So like, it's not like a duh.
Yeah. So, huge molecule. And if you want to know, if you took the DNA out of your cells and laid it end to end, how long that would be, I don't know. It's also a really big number. And when you say things like to the moon 3 times, it's an incomprehensibly big number to me anyway. It's 2 pairs of separate strands. They wind it around to form a double helix. I've given you some diagrams. We're going to go over this in detail. I'm going to have you draw one. But I'm not going to have you draw one now because I'm going to give you the last 20 minutes of class to go over your lab. And I have a few things on your lab. So first thing, grab your lab books.
Are they marked?
I have not written on the sheet, so you're going to keep the sheet in there. I have looked at them all, but I'm going to fully mark it after I see your quiz as well. So grab your lab book.
The form you sent out is for like finishing
If you didn't finish the lab, yeah.
Tuesday, right?
Yeah, 7:30.
Yeah.
Oh, you should know. That's why I couldn't find, that would make sense then. Okay, you're going to keep it now,
Okay.
but on Wednesday, the day of the quiz, you're going to leave it in class.
Okay.
Okay? Because you're going to put the quiz in it. Okay, thank you for letting me know though. That actually explains a lot.
When we didn't take the table of
Well, you're gonna get to take it home.
I know, but then in the table of contents, I forget to put in the other.
Yeah, the other labs.
Also,
No, but like I forget to like close it off. Cause I have like, cause my acid basis lab starts at 11, but I don't know what's gonna finish it, so I generally just forget to close.
Oh. I mean, the table of contents can have, doesn't have to go in order of pages. I don't think I actually put that in a criteria, because so you could also put in page 5, this. Maybe do something that makes sense.
Mr. Neufeld, we fill our questions on the physical paper.
Yeah, I'm gonna go through it for everybody, but yes. Yep. Okay, sitting down with your lab, I'm gonna go through a few things. Jojo, I don't think that's working. How about, I haven't seen one fair since I've seen both of you. Rock, paper, scissors.
What?
Jojo, rock, paper, scissors.
What?
One of you's going to move.
Huh?
Yeah, rock, paper, scissors. One of you's gonna come up here. Cause you're always fighting. Go.
We're not fighting, we're playing.
Yeah, then no, not right now. Rock, paper, scissors. Okay, right up there.
So how,
Yeah, no, come. Right up there. Let's go. Okay, number 1. Turn to this page of your data booklet.
So you can cover that.
I don't know what that is. Oh, I have it.
You'll only. I don't know if you stuck that one back. I'm not sure if you need to have it.
I'll see you.
What are you looking for? Finish it afterwards, right here. Pablo, we're gonna do this right now. You can finish that up after. Got your data booklet up? Okay. Let's take a look at the row with phenolphthalein. If it's a pH of 11, very basic, what color would you expect it to be?
Pink.
All together.
Orange.
Pink. Please don't call out nonsensical answers when you know they're nonsensical. So it'd be pink. If it was indigo carmine, what color would it be?
Probably
Less than red.
blue.
12? Less than 12?
No, it's blue.
How are you getting red? From indigo, do you have your data table out?
Yeah.
Have you found indigo carmine?
No, let me think.
Look on here.
Oh.
Yes.
If it's like if it's yellow and blue, would it just be a mix of both?
Yeah. So if I ask you something and I say a pH of 3 with methyl orange, not methyl orange, say a pH of 4 with methyl orange, you could say, oh, it'd be red yellow. What is red yellow? Orange. So if you say orange, that would be correct. If you say red yellow, that would be correct. If you said blue, because you thought red orange was blue, you would be wrong. So if you don't know your color joining, and I'm actually one of those that forgets that all the time, then just say red yellow. That's perfectly fine. Okay? But you would expect it to be a bit orange. So these show you the pH ranges of the different indicators. One of them you used in the lab, phenolphthalein, and it talks about in your lab that it turns pink from about pH 8 onwards. And so you start seeing pink in there. If I asked you for indigo carmine or asked you for one of the others, I would expect you to be able to read off of this what it would be if I gave you the pH or gave you what it was. What would grapes be in methyl red? Red. What would soap be in phenolphthalein? Pink. There's soap, little sud thing. There's pink, just the start of pink. Okay? So you have that table with you for your quizzes. The next part is a correction on your lab. So have your lab page out. But first, look at your observations. Which solution, A or B, went pink in phenolphthalein? Not the unknowns, but the first solution, A or B. Which one went pink in phenolphthalein? B. So which one's the base? B. In question number 3, it says solution B was hydrochloric acid. Does that make sense with your results? No. And what happened is I gave the lab the instructions to Ms. Stewart, and I didn't give her the questions or tell her to do it the other way around. So she just made it up and assigned them randomly, and they were different from the questions. So I want to correct this. Cross out where it says solution B was hydrochloric acid, solution A was sodium hydroxide. What is correct is solution B was sodium hydroxide. Misspelled there.
Do we need to write down the new one?
You can correct it in any way you want. You can just correct A and B, but I wanted to make sure it was written in properly, so I've written it like this, and I'm going to upload the new one to the Google Classroom, so if I print one off, it'll be corrected.
Wait, what do we cross out?
So where it says solution B was hydrochloric acid, you could just cross out B and write A, and for solution A, you could cross out A and write B.
Oh.
But I rewrote the whole sentence just to make it very clear. But you can correct it in any way you want. There's one other correction, and that is correct. Number 2, it says on a scale of 1 to 14, what the approximate pH of the new one is unknowns be. The old one was your solution, and the reason for that is in this lab, a long time ago, I did it at Stellies High School, and I said, bring in a solution, and then you get together with a bunch of people and you test each other's, and then that was to say the pH of yours. And it turns out we live at a boarding school, so if I say for you to bring in one, a lot of people don't have anything. Whereas if they live at home, they can go to their cupboard and go, oh, I'll take some of this or some of that, or this Drano from underneath the sink. And you don't necessarily have access to that, so I gave you 5 unknowns. So this should have been changed to what is the pH of the unknowns? You don't know what the unknowns are, so all I'm looking for is it above 7, below 7, or equal 7? Is it acidic, basic, or neutral to make sure you know what pH is with respect to those? So I have changed it slightly to reflect that. And I put there phenolphthalein indicator turns bright magenta. And I made it bright magenta just for fun. Yes?
So for unknowns, is that solutions,
A, B, C, D, E.
A, B, C, D?
Yeah. Good. So for your quiz, you will be either allowed a data booklet, or I will put the portion of the data booklet you need, which would be the indicator page, in your quiz itself. You will be allowed your lab book, and in your lab book, you should have the marking sheet. Not that that would help you in the quiz, but it should be there. And you should have the lab sheet that looks like this. If you don't have one, I printed some extra off, and they actually already have the corrections on them. You can grab one, and that way,
Yeah, can you pass me one?
there are a few more. Keep them in your lab book. And then after the quiz on Wednesday, you're going to leave your lab book with the quiz inside of it in class, because my favorite Christmas activity on December 24th, Christmas Eve, is to the good roaring fire and a purple or green pen, is to sit with my feet up and mark chemistry labs. It just fills my soul.
Not spend time with your kids.
Have you met my kids? Oh,
Work is awesome.
they're terrible, terrible people. Oh. I'll never forgive one of them either. I mean, one of them's birthday. You know the night where you have your last meal with the house, like coming up right now?
Yeah.
One of them was born on that day. Like, I'm trying to be with the house and you're going to be born? How dare you?
Which one?
Never looking ahead. Not time. Can't tell. Although if you're in Pyramid House, you're going to celebrate a birthday soon. It's not on the same day though, because the day changes slightly from year to year. But sometimes it is, yeah.
Wait, so if you had to choose, would you go with the house or for the birthday?
Oh, I had
You had to go.
the person who was giving birth, like, would be really upset if I wasn't there.
Are you complaining?
You gotta go.
What's the quiz about?
So, the quiz will ask you questions related to or identical to the questions of the lab sheet or about your observations. So, the quiz is open lab book. Not open people, not open internet, but it is open lab book. So if you bring all your observations and you bring answers to the questions, you have an open book that you're allowed to use. When you're done, you close it, put it aside, you're good. It'll be on Wednesday. I don't know if I'll be delivering it. It might be a BFA because I can feel this sickness taking hold. It started last night, and I'm thinking by Wednesday I might collapse. As a house parent, I have to schedule my days off because tomorrow they're full block classes like today, but Wednesday's classes are slightly shorter, so if we're going to miss something, maybe Wednesday to try and get better. However, I might still be here anyway because class first. So you are going to take your lab book home with you tonight. So you get to keep your lab book right now. Keep all the pages in it. Don't lose the pages. You might need all that. So you have time right now to answer the questions, ask me questions, clarify things, fix things. Your lab books are yours. Is there any lab books that are left? Let's see. Is anybody in Riley's house?
I know.
If you could, ask her to take it before she left. Anders, you haven't gotten your lab book. There you go. Let's get started. And no name on the front.
I'm not finished.
Lucas, you haven't even grabbed your lab book. Number one, put your name on the front. Okay?
50 still on the table.
Which ones were asked? Or
Yes, you don't have to ask. Remember.
The fourth question said we need to state each unknowns as a little bit, so I already did it in the second.
Yeah, so that was part of your part 2 observations. That's just answering the question.
Yeah, so can I like just want this one because I like made a.
Oh, you made a whole table. Yeah, that's the. Now, did you put a number and a title on your table? So you want like table 4. Da 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 da.
No, it's like that's for the questions to use answer.
Yeah, I know, but here, table 1, right? Table 2, table 3.
Huh?
Every table, table 4.
Oh.
And then in that question, you say, refer to table 4. There it is. There and then there. And then there.
I got so before I listened to it. I got that, which seemed more plausible. And
Yeah.
I googled online.
No! God, that looks like ChatGPT.
Yeah.
Yeah, the ChatGPT doesn't know.
It wasn't ChatGPT.
Have you looked up acid-base equations from our notes?
Does this smell like?
Oh shit.
Do that. I'll do it on the board if you can piece it. But
Oh.
it needs, what number is that for? Pardon? Water and,
salt.
yeah,
Okay.
yeah,
Isn't there? Oh my gosh.
yeah.
Oh my gosh.
Wait, we don't have to copy the other questions, do we?
You should.
No, there's on the corner
You
of
have to
the
copy down the question to fix the
Yep.
answer.
You have to write
One
it
thing.
full sentence.
The answer, but not the question.
You got 17.
all finally in notes. By what they use, you test it all. So you go. Lucas, Pablo, I'm seeing you guys turn. Nobody's lab books are open or working. Your lab books should be on your own table. Yeah, but you're not open or working. So turn around. Get them open. Start working. Julian, yours doesn't seem to be out at all. No, you have 20 minutes to
Basic
work.
or 5?
20 minutes.
It's
10
5.
minutes.
Why do you say
5
it's
minutes.
What time is it?
I started at
Because
12:40,
it
so you have 5 more minutes to work.
Salt?
Get working.
What is the answer?
Oh, you do.
It's
You
around
do.
7.
You can figure it out.
It's
Okay,
around 7.
there's no line.
What are the products of an acid-base reaction?
Put two back into the water. Also, it's around 7. 7 pH.
That's combustion.
Neutralization. Yeah, Julian? That's not writing, that's just loud.
I need to put the complete answer, right?
No, we're thinking combustion again.
That's like AB. What's AB?
So, if you, what does an acid have?
Acid. H.
Yeah.
H. H.
What does a base have?
OH.
He gave it.
Right.
OH. So H2O and AB.
Men's body kind.
And it would be Ba, because that's the metal,
Oh.
right? Now, you've done this,
Right now,
and you have all this left,
you grab your lap too.
So
and then you boil it away. What's the white residue?
BA. But how can I know what is it?
But you have to go look at what it is.
Okay, now you've got.
It's either, because for solutions A and B, you know you use HCl and NaOH.
Oh, we know? How
Yeah.
do we know?
It tells you in number 3. That's what we corrected.
Okay, quick question. Is this?
According to table 1, solution A was an acid. Oh, that would work because you're referring to a table. Yeah. You could also say the litmus paper turned red, therefore it's an acid. So those would be good. Yeah. You can refer to a result. You can refer to a table. Gentlemen, you should not be backed up. No, Julian, you just poorly. But
It's
this
just
is also
metallic
the
bond. It's a metallic bond.
Have you done all the questions?
So it'd be a zinc hydroxide plus sodium. Then it's
Results in water?
No.
Consider
You're
results
gonna
in
get a
water.
quiz on it. I'm not
Maybe
going
that's
to
what
help
it
you.
would be. Yeah.
So, but you get to bring in an example in which
I
you
think so,
write the
yeah.
answers.
Wait, can I just have another piece of paper?
And sodium what? Just sodium.
It has to be right.
Yeah.
Art.
Isn't that like a plastic?
We'll just go back
Laptop,
to our lab.
open, working. Where's your answers to questions? Laptop, open, working. The thinking only applies if you're thinking about what you're supposed to be doing. Yeah, let's go. Open it up.
Upside down.
Thomas to you.
Hmm? Yeah, but I would, that would be good, but at this point, you need your observations. That's one of the things I noticed is that you didn't have them for the last part. Probably get those down. And then you wanna do the answers to questions. So you got a fair chunk to do there before Wednesday's quiz. Yep. And you could stay until 1:15. Yeah, you could. Oh, you don't need lunch. Sorry,
If it already
I'm
actually
giving you
base, right?
senior privileges. Let's just say I want to see the answer to the first question. Yeah, first question.
Oh, it was.
Or your observations done.
It's if it's a base. Yeah. No, it's not.
Also, if you're curious, you don't need this for your lab, but of the unknowns, solution A was lemon juice. You may have smelled that. Solution B was vinegar. Solution C was Perrier, which, depending on how long it sat, may have been acidic or may have been neutral. D was household ammonia, and E was Alka-Seltzer, dissolved in
So
water.
can we use that in our answer?
I'm just telling you to see, so you can look up if it matches if you want. You don't need it for your answers, because you have your tables of observations.
Yeah.
And sodium from the form of a gas.
Sorry, wait.
Is it possible? Yes. In this lab? No.
Okay, because I changed my answer like 10 times. I think
Which one are you answering again?
7. Where's the gas? Because when you add
Oh, we did that outside. There, remember? And we lit it. Sorry, I thought you were actually answering a different question. When we dropped, so actually, okay.
So it doesn't result in water?
Yeah, sorry, I thought you were doing the acid base one.
You got no turn.
Zinc plus HCl goes to,
Isn't it
Um,
in base?
now,
With lemon
zinc is a metal, but it can become +2. Which one of these can be negative?
Chlorine.
Chlorine will always be negative. So there.
I thought that it was in
What's
base.
left over?
Hydrogen gas. But if that's in my lab, it says that base reacted with the same.
Pardon? Your lab, if it says base reactive with zinc,
Or my
is
results.
wrong.
My results.
Yeah, it's an anomalous result then. Maybe you saw bubbles? Show me.
Also, for Perrier, instead of the acid or base, do you think aquatic and
The Perrier, if fresh, would be acidic, but if it's sat and the bubbles came out, it can go to neutral and even basic. So it actually just depends on your results.
Yeah, okay.
Yeah.
Solution A was the acid. Got Perrier was base.
Exactly. You need to write a little slower, practice being a little neater, but then keep going. Okay?
Can I switch my things? Cause
No, so just it.
or no, cause it's
The one that reacted with the zinc, did it turn phenolphthalein pink or colorless?
colorless.
So that means it was an acid. What solution was it? A is an acid. That's not base. No, remember, look over here.
No, but in mine, it reacted with no, the one that reacted turned hot pink. Sorry, in phenolphthalein.
Oh, okay.
So-
And that is just an anomalous result.
So I think I did that on a separate day. So I probably switched it.
Either you switch the solutions, it may be, sometimes students see bubbles that aren't there because they're expecting to see them, a bit of a placebo effect. So if that happens, you know, because the test wasn't, especially with the solutions, it wasn't definitive. Sometimes, the reason I include the test is sometimes the zinc is good and it shows, and sometimes, I don't know if it gets coded, it just doesn't work that well. And on those 2 days, it didn't seem to work that well. So don't put a lot of faith in the zinc results, but phenolphthalein and litmus work every time.
Alright.
Yeah.
So just like, can I just say that like, oh, I believe that they were switched because.
Yeah, or you can just say, it reacted according to my observations, but you know, because I demoed it in front of the class, that it requires an acid. That's why I did the zinc test in the 6 molar HCl to make sure we had a really good version of it.